Okay, so welcome back to this next video in which we are discussing high-density lipoprotein and the reverse cholesterol transport process. Okay, right, so we've had rather a large deviation into ABC transporters, which I think is worth it because we're going to see a lot of them in this process, okay? And the one that we started off with was this ABCA1, which was a full transporter which was on the surface of these hepatocytes and these enterocytes. Okay, so this is ABCA1. And basically it's going to use ATP to transport cholesterol and also phosphatidylcholine molecules onto the apolipoprotein A1. Okay, so here is our apolipoprotein A1, remember? Okay, which the hepatocyte or enterocyte has just produced. So I'll colour in the ApoA1 here in turquoise. We'll have ABCA1 in red. Okay, so this is ABCA1. And I'm sorry, I didn't mean to imply that the cholesterol molecules and the lecithin molecules will be transported at the same time. They'll be transported separately. Remember I said that the curious thing about these ABC transporters is that they are able to bind and transport a wide variety of different molecules across the cell membrane. They're not specific for a specific molecule. Okay, they have very broad specificities. Okay, so we'll be transporting lecithin or phosphatidylcholine molecules and also cholesterol molecules out of the cell and onto the apolipoprotein A1 here. Okay, and what this will allow you to form is these small discoidal HDL molecules. Okay, so basically these consist of a phospholipid monolayer, like so. Okay, but then with no hydrophobic core, so there's no cholesterol esters yet within this. Okay, so you don't have any sort of volume in there, so it's just a flattened disc basically. Okay, so here is the apolipoprotein A1. Okay, right. So let's now talk about other things that are going to be produced by hepatocytes and put into these discoidal HDLs. Okay, so something we're going to come across very soon is an enzyme called lecithin cholesterol acyltransferase. Okay, so I'd like to put this in now, and this is going to be produced by hepatocytes and put into the discoidal HDLs which are going out into the blood. Okay, and there are other things as well which we'll come to when we need them, but this is a very, very important one, so I'd like to mention it now. Okay, so this is lecithin cholesterol acyltransferase, and we're going to be using this very soon. Okay, and for short, lecithin cholesterol acyltransferase is often abbreviated to LCAT. So L for lecithin, C for cholesterol, and A for acyltransferase is abbreviated to AT. Okay, right, so here we then have the apolipoprotein A1 in turquoise, and we then have this phospholipid monolayer, and these phospholipids have come from the ABCA1, which is transporting out mainly lecithin, but it might be able to transport other lipid molecules as well, such as other phospholipids. Okay, and it will also be transporting out cholesterol molecules, free cholesterol molecules, which will implant into the phospholipid monolayer as well. Okay, so we'll denote the free cholesterol molecules here in blue, and then let's also colour in those polar heads of the phospholipids. Okay, so here in orange, these are the polar heads of the phospholipids. Okay, so we've now created what is called a small discoidal HDL molecule. Okay, so a small discoidal high-density lipoprotein. Some people will also call them nascent discoidal HDL proteins. Okay, right. So, these things then are going into the blood from hepatocytes and also from these cells of the small intestine. Okay, and these are the things which are going to be taking the excess cholesterol from cholesterol overloaded peripheral cells. Okay, so now it's time to meet the cholesterol overloaded peripheral cell. Okay, so let's now go to our peripheral cell that has far too much cholesterol within it. Okay, so this is some cell tissue in somewhere in the body, basically. So this is a peripheral cell, and it's suffering from having too much cholesterol. So I'll draw a little picture of the cholesterol molecule within it. Okay, so we've got far too much of this. So here is the B ring, which is this 6-membered carbon ring. Here is the C ring here. Then we've got the D ring, which is this 5-membered carbon ring. We've got methyl groups coming off up there and there. We've got an alcohol group down here. Okay, and then we've got we've got a double bond there, and then the side group up here, like so. Okay, so we've got far too much cholesterol within this cell. So, what signaling pathway is this going to activate within the cell that is going to help you get rid of this excess cholesterol? Well, basically, it's going to activate what are known as liver X receptors, but it isn't cholesterol directly which activates the liver X receptors. Instead, it is things called oxysterols. Okay, and I want to just tell you about what oxysterols actually are. Okay, so these are molecules which will go up in the cell when you've got too much cholesterol. Okay, so they're derivatives of cholesterol. So if you've got a very large amount of cholesterol in a cell, then you'll start getting oxysterols appearing. So let me give you some examples of oxysterols that are found in different portions of the body. So the first oxysterol I'd like to introduce you to is something called 24S. Okay, and then 25, and then it's epoxy cholesterol. Okay, so epoxy and then cholesterol. So let me talk you through the structure of this then. So let's draw a little picture. Okay, so for this we need to understand the naming of cholesterol again. So we need to remember our numbering of the different carbons within the cholesterol molecule. Okay, so here is the A ring of our cholesterol molecule. Here is the B ring. Here's the C ring. And if you remember, 24 and 25 will be up in that 7-membered side chain that comes off carbon 17. So remember, the way that you number the carbons within a cholesterol molecule are, this one is number 1, this one's number 2, this one's number 3, this one's number 4, this one's number 5, this is number 6, 7, 8, then 9, then we have 10 over here, 11, 12, 13, 14, 15, 16 and 17, but of course we haven't finished the cholesterol molecule yet. Okay, so we also have these two methyl groups here. We have a double bond between 5 and 6. This one then is 18 here, this one is 19, and then we have this side chain that comes off up here, okay, like so. And in fact, I actually don't think I ever told you the way that you adapt the numbering system for cholesterol. I do apologise. Right, getting confused between videos. Right, so we know the numbering system for our steroid structure. How do we adapt it for cholesterol? Well, basically, we continue on from 17, and then we call this methyl group up here 18. We call this methyl group up here 19. So the carbon at the centre of that methyl group is called 19. And then as far as the side chain up here is concerned, let me draw out this side chain here. Okay, this carbon then is called 20. This one's called 21. This one's called 22. This one's called 23. This one 24. This is 25. This one then 26. And this one then 27. So basically, what you do to turn cholesterol into 24S25 epoxy cholesterol is you're going to make a link between this 24th and this 25th carbon, and it's going to be a single oxygen atom sitting between them, basically. So you're going to form a covalent bond between this carbon and this oxygen, and this carbon here and this oxygen. And when you have an oxygen sitting in between two carbons like this, forming a triangle, okay, that's known as an epoxy group. So that's why this is called 24S25 epoxy cholesterol, because we've got the epoxy group between the 24th and the 25th carbons. Don't worry about the S, okay? That just refers to the optical isomerism of the whole thing. Okay, but the basic concept is there, that you've got this epoxy group here. Okay, so 24S25 epoxy cholesterol. Okay, now this is a really important oxysterol that you form within hepatocytes. Okay, so I said we were talking about peripheral cells. I didn't say we were talking about a hepatocyte. Okay, so this isn't really going to be the oxysterol potentially that you're going to be making in peripheral cells when you've got too much cholesterol. However, later on in this process, we're going to see that the hepatocytes will end up with too much cholesterol, and this is the oxysterol they will form, so knowing about this one will be important later. Okay, so let's see some more examples of oxysterols. Okay, so again, they're just simple modifications of cholesterol where you have oxygen atoms, basically, within the structure. Okay, so another example of an oxysterol is 24-hydroxy cholesterol, and this one's very simple. All you need to do is put an alcohol group coming off the 24th carbon of the cholesterol structure. Okay, and we've got much more space now, which is much better. Okay, so let me draw out the cholesterol structure here. So here's the A ring, here is the B ring, okay, here is the C ring, and then off the C ring we've got the D ring, the final 5-membered ring. Okay, we've then got the methyl group here, the methyl group here, the double bond here, and the alcohol group of carbon 3 down here. We've then got our 7-membered side chain up here, and all we need to know is which carbon was the 24th. Well, remember, this was carbon 17, this was carbon 18, this was carbon 19, this one was 20, 21, 22, 23, 24. So we're basically going to stick an alcohol group off that, and that's how we get 24-hydroxycholesterol. Now, this one is extremely important in the brain. Okay, so this is a very important oxysterol that's formed within the brain. So one final example, which is found less selectively in a single place, is 22R-hydroxycholesterol. Okay, so 22, and then you have R, which just again refers to the optical isomerism, and then we have hydroxycholesterol. Okay, so this is again very easy. We've just got a alcohol group coming off this 22nd carbon. So where was 22nd? Well, here was 20th, 21, 22, 23. So instead of the alcohol group coming off the 24th here, you're now going to have it coming off the 22nd here. Okay, so here is the alcohol group there. So take that off, put this one on instead. You've now got 22R-hydroxycholesterol. Okay, right. Those are examples of oxysterols. So these molecules will go up within this peripheral cell which is overloaded with cholesterol. So let's say this is our peripheral cell again. Here's its nucleus. And it's now starting to get oxysterols being formed within its cytoplasm, basically. Okay, now, what's going to happen is these oxysterols are going to activate nuclear transcription factors, okay, called liver X receptors. So I now need to discuss liver X receptors with you. Okay, so basically there are two main types of liver X receptor, and they are often abbreviated to LXRs for short, liver X receptors. So the two different types are liver X receptor alpha, and then we also have liver X receptor beta. Now, liver X receptor alpha, as a general rule, is found within the liver. So it's found within hepatocytes. So again, we're not looking at this pathway yet in hepatocytes. We're looking at it in a general peripheral cell. However, later we will be looking at it within hepatocytes. So that will become important later. Liver X receptor beta is found pretty much everywhere, basically. So in peripheral tissues. So this is the important one for us now. Liver X receptor beta. Now, basically, liver X receptor betas, and in fact liver X receptor alphas as well, so I'll just from now on just refer to them as the liver X receptors because they both do exactly the same thing. Okay, they dimerize with another type of receptor which is also in the nucleus. Okay, so I don't know if I've emphasized that enough. These always stay within the nucleus, so they are what are known as a nuclear transcription factor. Okay, so some transcription factors do go into the cytoplasm, not liver X receptors. They always remain within the nucleus. Okay, right. So what happens is the liver X receptors, they will dimerize with another receptor called the Retinoid X receptor. Okay, and they will do this whether they have been activated or not. Okay, so at the moment the liver X receptor is inactivated, let's assume. It's going to be activated by the oxysterols, but at the moment it's inactive because it will have an activity even when it's inactive. So I want to describe to you first what these do before they have got the oxysterol bound to them. And then we'll move on to how that function changes when they do indeed have the oxysterol bound to them. Okay, so at the moment we have our liver X receptor, which I'll outline in red here. So let's assume this is our liver X receptor here. Okay, and I'll just abbreviate it to LXR for short. Okay, and it's in the nucleus of our peripheral cell. Okay, and it is now dimerized with a totally different receptor, which is known as the Retinoid X receptor. Okay, and I'll tell you what the ligand for the Retinoid X receptor is at the moment, in a moment rather. It's obviously a retinoid, but I'll tell you exactly which one. Okay, so this is the Retinoid X receptor. Okay, and for short, the Retinoid X receptor is often abbreviated to RXR. Okay, right. So, even when neither of these two receptors have their ligand bound to them, what will happen is this complex will be formed and it will bind to promoter regions on DNA. Okay, so let me show you some DNA now. So, here is a piece of DNA. Basically, upstream of all genes in the eukaryotic cell, okay, you find what are known as promoter regions. So let's say this portion of DNA here is a gene. So I'll highlight this in green here. And upstream of absolutely every single gene in a eukaryotic cell, but not in a prokaryotic cell, in a eukaryotic cell, there is a region known as a promoter region. Now, this promoter region controls the expression of the downstream gene. It itself is not translated into protein. Okay, so it will not be read by a ribosome and converted into a sequence of amino acids. However, its role is very important. It's going to control how much of the product of this gene you actually create. Now, how does it do that? Well, basically, if you're going to actually make the protein that is associated with this gene, what has to happen is the RNA polymerase II enzyme has to come and bind to your DNA. It has to open your DNA, so it has to split apart the two strands of DNA. It then has to work its way along the DNA and produce a piece of complementary mRNA, which can then go and be translated into a protein. Okay, so where does the RNA polymerase II bind to? Well, basically it binds to this promoter region here. So RNA polymerase II will come in, it will bind to the promoter region, it will open up the DNA, it will work its way along the coding strand of DNA, producing a piece of mRNA, which will then go out of the nucleus to the ribosome and be translated into protein. And so, the promoter region therefore controls how much of the gene is going to be made, because if the promoter region has a very high affinity for RNA polymerase II, then RNA polymerase II will bind here all the time, okay? Therefore, you'll get a huge number of mRNAs being produced, and therefore, you'll get a huge amount of the protein being produced. Okay, whereas if this promoter region has a very low affinity for the binding of RNA polymerase II, RNA polymerase II will bind there hardly ever. You'll get hardly any mRNA being produced, and therefore you'll get hardly any protein being produced. Okay, so depending on the affinity that the promoter region has for the RNA polymerase II, you're going to get different amounts of the protein associated with this gene actually being produced. Now, a transcription factor then, we move on to the next thing. What does a transcription factor do? So basically, a transcription factor is any molecule which can bind to promoter regions upstream of certain genes, okay? So a single transcription factor will bind to a plethora of promoter regions upstream of genes. It won't bind to all of them necessarily, but it will have a good 100 or so that it binds to, okay? And what it will do is it will change the affinity of that promoter region that it binds to for RNA polymerase II. Okay, now at some of the promoter regions that the transcription factor binds to, it will increase the affinity of the promoter region for binding RNA polymerase II. And at others that the transcription factor binds to, it will decrease the affinity of that promoter region for binding to RNA polymerase II. If it increases the affinity of that promoter region for binding to RNA polymerase II, then RNA polymerase II will bind more often, you'll get more mRNA being produced and therefore more protein being produced. The transcription factor has hence enhanced the expression of the downstream gene, and we will call that a transcriptional enhancer. Okay, for that specific promoter region and that specific gene downstream of that promoter region. If, on the other hand, the transcription factor binds to the promoter region and stops it from binding to RNA polymerase II, or decreases the affinity with which RNA polymerase II binds there, then you will get RNA polymerase II binding there less often, you'll get less mRNA being produced, and therefore less protein being produced. We will call that a transcriptional repressor, basically. Okay, so transcription factors can enhance the transcription of certain genes and repress the transcription of other genes, so they alter the expression of genes, basically. Okay, so what's going to happen is when you've got liver X receptor and the retinoid X receptor heterodimerized like this, it's called a heterodimer because you have two different proteins dimerized together. Hetero means different. Okay, so you can also refer to this as an LXR, liver X receptor, RXR, retinoid X receptor, heterodimer. Okay, which just tells you exactly which two proteins are within the heterodimer. Okay, right. Now, when neither of them have their ligands bound, what happens is they bind to a third protein called the Corepressor. Okay, so this is called a Corepressor. And this complex of the liver X receptor, retinoid X receptor, heterodimer with the Corepressor is then going to go and bind to the promoter region of certain genes and stop them from being transcribed. Okay, now which genes am I talking about here? Well, I'm talking about the genes which are going to help you release cholesterol from the cell, basically. Okay, so what's going to happen is that this will repress transcription of certain genes. Now, I haven't told you which genes it's going to enhance the transcription of when the ligands are actually present yet, so I can't tell you yet which it's going to repress, okay, but you will see the significance of this later. Okay, so when there are neither ligands for retinoid X receptor or the liver X receptor present, then a corepressor will bind to the liver X receptor, retinoid X receptor heterodimer, and this will then go and repress the transcription of a whole bunch of genes which are going to be involved in removing cholesterol from the cell, basically. Okay, right. On the other hand, when these oxysterols go up within the cell, what will happen is oxysterols will bind to the liver X receptor. Okay, you're also going to need the ligand for the retinoid X receptor to bind as well. So let me now discuss what the ligand for the retinoid X receptor is. So basically the ligand for the retinoid X receptor is something called 9-cis retinoic acid. Okay, now to understand the structure of this, what I firstly want to show you is the structure of Alltrans retinoic acid, okay, which is often abbreviated to ATRA for short. So the A is for all, the T is for trans, the R is for retinoic, and the A is for acid, so ATRA. Okay, so let me start by showing you the structure of Alltrans retinoic acid, and then we can adapt Alltrans retinoic acid into 9-cis retinoic acid. So, basically, these are derivatives of vitamin A. They have a very similar structure to vitamin A. Vitamin A has an alcohol group on the end rather than a carboxylic acid group, but apart from that, it's the same thing. Okay, right. So you have this 6-membered carbon ring here, okay, and we're drawing a skeletal formula. And then off this carbon up here, you have 2 methyl groups coming off that. You then have a double bond here, and you also have a methyl group coming off down here. Then, off this carbon here, you're going to have a long tail, basically, okay? And you have double bonds every other bond, okay? So we have single bond, double bond, single bond, double bond, single bond, double bond, and this continues on for 4 repeats, basically. Okay, then a single bond and a double bond, so only one more. Okay, then the final group that you have right on the end is then a carboxylic acid group. Okay, and carboxylic acid groups always look a little bit weird when you draw them skeletally. But remember, this is the carbon of the carboxylic acid group with the double bond to the oxygen and the alcohol group coming off here. Okay, right. Then we also have a methyl group coming off here and also a methyl group coming off there as well. Okay, so this is the structure of Alltrans retinoic acid then. Okay, so let's label up the different carbons within this structure then. So you start off with this carbon here. You call that number 1, you then go round carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, you then go down the tail, carbon 7, carbon 8, carbon 9, carbon 10, carbon 11, carbon 12, carbon 13, carbon 14, carbon 15. Okay, right. Then where do you go after you get to carbon 15? Well, you go back to this one up here first. This is carbon 16. Then this one's carbon 17. Then this one's carbon 18, carbon 19, and then carbon 20. So it's all pretty common sense. They don't sort of dot all over the place in some nightmare way. Okay, right. So, I want to discuss what we mean by Alltrans here. Okay, this refers to the EZ isomerism of these double bonds between these carbon atoms that you've got. So all of these bonds here, these double bonds that we have between 2 carbon atoms, basically the 2 carbons that come off, that are involved in the double bond, have 2 groups coming off them. They have a hydrogen atom coming off them. Okay, so if we look at this carbon, for instance, 3 of the bonds are to other carbons, so the other one is to a hydrogen. And then the other group is some important group, which I'll call R. So for instance, this portion here. Okay, then off the other carbon, if we look at this one here, you've then got another important group coming off, which we'll call R', and then you also have a hydrogen. Now, there can be two conformations of this, basically. Either you can have the conformation that I've shown here, which is the trans conformation, okay, also known as the E isomer, or you can have this conformation, like so, which is where the two hydrogens are on one side and the two important groups are on the same side. Okay, so trans means on the opposite side, basically, and you can see that these R and R prime groups are on the opposite side. When they're on the same side, this is called the cis isomer. Cis means on the same side. It's also called the Z isomer. Z just means, well, it's for zusammen in German, which means together. E is for entgegen in German, which means opposite. Okay, right. So, basically, when we say all-trans retinoic acid, what we are referring to is the fact that all of these double bonds here, every last one of them, is in the trans formation, and that's demonstrated by the fact that this group here is on the opposite side to this group here. So this structure does show you that they're all in the trans conformation. If one was in the cis conformation, you'd have to draw the important group coming off down here, like so. Okay, so let me just put a sort of dotted line like that. In fact, I'll put it in colour. If this was in the cis formation, this important group would have to come off down here like so, to show that it was in the cis formation. So that's how this structure shows that it's in the all-trans form. What we're now going to create is 9-cis retinoic acid, and this is this molecule where just one of these double bonds is in the cis formation. And it's this double bond that comes off the 9th carbon here, between the 9th and the 10th carbon. So what we now need to do is flip this group round so that it's in the cis conformation and it comes off down here. Okay, and that will create us 9-cis retinoic acid. So let me now draw the structure of 9-cis retinoic acid for you then. So, we'll start off with the 6-membered carbon ring, which hasn't changed in the slightest. Okay, and you'll notice that all that's changed about this molecule is that we've changed the EZ isomerism, we've changed the stereoisomerism, and you might think, oh gosh, how trivial. Okay, but remember, enzymes are specific for stereoisomers. They like certain EZ isomers and they like certain optical isomers. It won't work with a different EZ isomer, basically. Okay, it's specific for the shape of the molecule. Okay, so we have a double bond here, we then have a methyl group coming off here, and we then have our long tail here, where we have these alternating double and single bonds. And now, this double bond here between the 9th and the 10th carbon, this is in the cis conformation. So now I'll draw it coming off like this. Okay, then what we need to have is our double bond again. Okay, so we'll have our double bond here, then we'll have our single bond, then our double bond, and then our carboxylic acid group down here. Okay, like so. So remember, we have these 4 alternating double and single bonds, and then we have the carboxylic acid group at the end. Okay, and we have a methyl group coming off this carbon here, and also a methyl group coming off this carbon here. Okay, and this structure that I've now drawn for you here then is 9-cis retinoic acid. Okay, and this is the ligand for the retinoid X receptor. Okay, so when both 9-cis retinoic acid and also the oxysterols are present, what that will trigger is a change in conformation, basically, of our liver X receptor, retinoid X receptor dimer. Okay, so here is the dimer again, and now both of them have got ligands bound to them. So here is the oxysterol, which is bound to the liver X receptor, and here is the 9-cis retinoic acid. Its structure's been demoted somewhat. It's now being represented as a Easter egg kind of shape, but that's what we're doing. Okay, so here is the liver X receptor in red. Okay, now bound to the oxysterol. Okay, and here in green, this is the retinoid X receptor bound to our 9-cis retinoic acid. Okay, which I'll denote in orange. Okay, and once you've got this heterodimer with the ligands bound, it will no longer bind to the corepressor. Instead, it will bind to another protein that's called the coactivator. Okay? So basically, when the oxysterol goes up, the oxysterol will bind to the liver X receptor. You'll then get this other ligand 9-cis retinoic acid binding, which, as long as you're not vitamin A deficient, you will have some of this within your cells. Okay? And then what you get binding also is the coactivator here. So this is the coactivator. Okay? And this complex will bind to the same promoter regions now, but instead of blocking their expression, okay, instead of repressing their expression, in decreasing the affinity of RNA polymerase for binding to that promoter region, now they will enhance the expression. They will increase the affinity of RNA polymerase for binding there. So they're going to increase the expression of certain genes that are now going to help us remove this excess cholesterol from the cell. Now, what are these genes? Well, there are two that are really important in peripheral cells. The first is ABCA1, which we've already seen. It's this full transporter, which is an ABC transporter, which can transport cholesterol and lecithin out of the cell. In addition, there's another one, basically, which is ABCG1. Okay? And this is another ABC transporter. Now, ABCG1 is actually a half transporter. So it's not a full transporter, it needs to dimerize, and it will generally form homodimers. So two ABCG1 proteins will join together to make a functional ABCG1 transporter. So let me just remind you of what a half transporter is. Okay, so you have your amino terminus here in the cytoplasm of the cell. You then have your one and only nucleotide binding domain here, or your ATP binding cassette. Then you have your one and only transmembrane domain, consisting of these 6 membrane-spanning alpha helices clustered together, and then you have the carboxylic acid terminus down here. Okay, so this is your one transmembrane domain. And what will happen is these will homodimerize, okay, to form an ABCG1 transporter. And these can also transport cholesterol molecules and also lecithin molecules out of the cell. Now, what's the significance of this? Well, basically, now's the point where our nascent HDL discs will come into play. Okay? So here is our cell, okay, which is completely overloaded with cholesterol. Basically, it will have these 2 transporters in its membrane, which are just going to be demoted to being represented as balls, which is the way that people usually represent them. Okay, one of them is ABCA1, so the ATP binding cassette transporter A1, sorry, and then one of them is ABCG1, the ATP binding cassette transporter type G1. Okay, so let's colour them in different colours. So here we have ABCA1, and here we have ABCG1, okay, and now the small discoidal HDL is going to come over, okay, so here is our disk of HDL, okay, which consists at the moment of a phospholipid monolayer with apolipoprotein A1 within it. Okay, and also remember that important enzyme, which is we're going to see the function of very soon, lecithin cholesterol acyltransferase. Okay, and what's going to happen is the apolipoprotein A1 will bind to the ABC transporter, either one of them, it doesn't matter. Okay, and what will then happen is that the ABC transporter will transport cholesterol and also lecithin molecules into the HDL molecule. And now what will that do? It'll make it bigger and bigger and bigger. It will mean that you've got more and more lecithin molecules making up your phospholipid monolayer, and you've also got more and more free cholesterol molecules making up your phospholipid monolayer. However, this will still lead to a discoidal HDL molecule. And because both free cholesterol molecules and lecithin molecules are stored within the phospholipid monolayer, not the hydrophobic core. So we're still going to have nothing within our hydrophobic core. Okay, so we're just going to make a bigger and bigger disc. That doesn't sound right. Okay, so what's now going to happen is the enzyme lecithin cholesterol acyltransferase is going to come into play now. Okay, so this is lecithin cholesterol acyltransferase, or as I said earlier, lecithin cholesterol acyltransferase is often abbreviated to LCAT. Okay, and basically this is going to use free cholesterol molecules and also lecithin molecules that we have present within the phospholipid monolayer to produce cholesterol esters. Okay, so here is the lecithin cholesterol acyltransferase. Now what's it going to do? Well, it's going to take a lecithin molecule which is in the phospholipid monolayer of our discoid or HDL particle. Okay, so here is our lecithin molecule. Now remember, the other name for lecithin is phosphatidylcholine. Okay, so let's just revise our structure. Here we've got the phosphate group. Okay, here this vertical line, which I'm now colouring in green, that represents the glycerol molecule. And then we've got two long-chain carboxylic acids esterified to the first and second alcohol groups of the glycerol molecule. Then, attached to the phosphate group of the phosphatidic acid molecule, we've then got choline to create us phosphatidylcholine. Now, basically, we're going to also take a free cholesterol molecule, so I'll draw my free cholesterol molecule here. Okay, so here is the B ring, here is the C ring, which is another 6-membered ring, and then we have the D ring here, which is the 5-membered ring. We then have our funny 7-membered side chain up here, the methyl group here, a double bond here, and an alcohol group here. Okay, right, so this is our cholesterol, and basically what we're going to do is transfer an acyl group from the lecithin molecule onto the cholesterol molecule. Now, what is meant by an acyl group? Well, basically, when you have a carboxylic acid which we are esterifying onto an alcohol group, you don't actually add the entire alcohol group on. Let me explain this. Okay, so remember, when we esterify a carboxylic acid, shown here, onto an alcohol group, we take the alcohol group off the carboxylic acid group, we take the hydrogen off the alcohol group, and we then attach the carboxylic acid carbon onto the oxygen of the alcohol group, like so, to form the ester link. Okay? Right, so we don't actually attach on the full carboxylic acid molecule onto the alcohol. Instead, we attach on this portion that I'm now circling in green here. And this group that we're attaching onto the alcohol group that is circled in green is called the acyl group. Okay, right, so that's why this is called an acyltransferase, because what we're actually going to do is take the acyl group that's attached to the second carbon's alcohol group of the glycerol molecule. We're going to take that acyl group off there and return this alcohol group to an alcohol group, and we're going to transfer it onto the alcohol group of the cholesterol molecule here, and hence we have transferred an acyl group from lecithin to cholesterol. So that's how this name, lecithin cholesterol acyltransferase, makes complete sense. So what we're going to therefore produce is a lecithin molecule that now lacks the long-chain carboxylic acid that should be attached in that second position here. Okay, shown here. So here is the choline molecule. Okay, here is the phosphate group, shown in pink here. Okay, here is the glycerol molecule, shown in green. And then here is the long-chain carboxylic acid attached to the alcohol group of the first carbon of the glycerol molecule, but we are now missing the one attached to the second carbon. This is now known as lysophosphatidylcholine, which I'll abbreviate to PC, or some people will even call this lysolecithin. Okay, right. The other product that you then get is a cholesterol ester. Okay, so I'll just draw up quickly out my cholesterol ester. So here is my A ring. Okay, then I've got my B ring here. Okay, then my C ring. And then my D ring coming off over here. And I've got a methyl group coming off carbon 10 there, a methyl group coming off carbon 13, and then this 7-membered side group coming off carbon 17 up there. Then I've got a double bond there, and now off this alcohol group I've got a long-chain carboxylic acid esterified here now. And to try and sort of parallel the two pictures, which is difficult because one's drawn as a cartoon, one's drawn as an actual structure, I'll put this as a sort of orange line there. Okay, so we have created a cholesterol ester here. Okay, so what will happen is the lysophosphatidylcholine, this will remain within the phospholipid monolayer of the HDL particle. Meanwhile, the cholesterol esters which we are forming, those will be going into the hydrophobic core. So what's going to gradually happen to our HDL molecule is that it's going to go from being discoidal in shape to being spherical, and now it looks more like the thing that we actually described right at the start when we were just discussing the core structure of an HDL molecule. Okay, so here is our apolipoprotein A1. We have our lecithin cholesterol acyltransferase here, also sitting in the phospholipid monolayer, force of habit there. Okay. Then we'll have many phospholipids sitting in the phospholipid monolayer here, with cholesterol molecules that are free interdispersed amongst them. And in the core we'll have cholesterol esters. Now you might be wondering, well, how do you gain more apolipoproteins? Because remember, HDL molecules, they didn't just have a single apolipoprotein in most cases. Well, the apolipoproteins are being produced by the liver and put into the blood, so they can just, join the HDL from the blood, okay? And as the thing grows, which will happen gradually because it will go round more and more cells in the body, it will take cholesterol from them by binding to their ABCA1s and ABCG1s, which are being expressed because of the liver X receptor interacting with oxysterols, and the HDL will gradually grow, it will get bigger and bigger and bigger, it will balloon up, and then more apolipoproteins can join basically and form this bigger and bigger structure. Okay, right, so we'll end there with one last thing to say before we do end. Okay, and we're only ending for a break, we're not finishing yet, we've still got a lot more to do. Okay, the one last thing I want to say is basically when the two ligands for the liver X receptor and the retinoid X receptor were not present, okay, so you didn't have both of them. You might have had the 9-cis retinoic acid, but you didn't have the oxysterols. Then you would have had that repressing complex, and this would have been stopping the production of the ABCA1 and stopping the production of the ABCG1, so that you were not getting the release of the cholesterol and the lecithin onto HDL particles, basically, okay? And that's good, because if you haven't got liver X receptor activation, it means that you haven't got oxysterols present in a high level within the cell, which means that you haven't probably got a high level of cholesterol, therefore you don't want to be getting rid of your cholesterol, okay? Remember, cholesterol is essential for the cells to live. It's an essential part of the cell membrane. They only want to get rid of it if they've got too much of it. Okay, right, so we'll call it there for this video, and we'll continue our discussion of the reverse cholesterol transport process in the next video.